And we're live. Good morning, everyone out in YouTube land. So, we have been talking about the movement of molecules across the plasma membrane. And the whole point of this is that the phospholipid bilayer provides a very significant barrier to anything with a charge on it. And if we were to think about stuff a cell needs to take up, well, a bunch of it is going to have a charge. I mean, not the least of which is glucose. All cells have to take up glucose in the bloodstream in order to actually carry out metabolism, which we'll be getting into. Glucose has got a pretty strong charge to it. So all of the different ions that you need for, we're going to be getting into the transmission of a neuronal signal here in a bit. All these things need to be brought into the cell. Additionally, a cell is able to operate because it has a asymmetrical distribution of different kinds of things. It can accumulate some stuff and it can dump stuff out. So the membrane serves as this barrier and compartment. And when we begin working our way through the different kinds of movements, we start out first with diffusion, which is just simply a physical phenomenon of the movement of molecules from an area of higher concentration to an area of lower concentration. This really is just an absolute fallback from the second law of thermodynamics, that systems have organization, i.e. a greater collection of stuff, and then over time that collection is going to dissipate. And so we've got this thermodynamic energy going on in terms of the accumulation of different molecules. If you accumulate stuff, it requires energy. You can dissipate that energy in a way, or extract that energy by allowing that material to actually then go back out and be resolved into equilibrium, so you don't have a concentration gradient. So that's diffusion. And specifically, the diffusion of water is a critical part of cell metabolism or cell operations, in that this is osmosis. And we laid out how we can think about the diffusion of water very much that water is following its concentration gradient, i.e. if you have a solution that has dissolved stuff in it, it's going to have actual less water molecules. And so the more dissolved solute you've got, the lower the water concentration is. And so water is moving along its diffusion gradient. The other way of thinking about this, of course, is that water always moves towards salt. So diffusion then is, in a sense, of an outcome of the accumulation of these ions on one side of the membrane versus the other. So diffusion requires this semi-permeable membrane, the cell membrane. So then we can subdivide, and let's see, where do I have room on my whiteboard here? We can subdivide diffusion, or the passive transport, in the following way. And this is a good way to kind of keep this side of the transport system in mind. So we've got the passive transport on this side, active transport on that side. So we've got then just plain passive transport of water, diffusion like that. We can also throw into this things like sterols. They can diffuse back and forth across the membrane as well. So then we have the mechanism of taking ions up, and we can think about either a passive transporter, and the key point is, on this side of the chart, we're using an existing gradient. And a passive transporter, and again, if you didn't see the video, well, this is like the experiments that Overton was doing, where they were looking at the glucose passive transporter, and that transporters are going to be both saturable and they are very specific. We also have another passive transport system that is referred to as not a transporter, but a channel. And the difference being that the channel is essentially a tube. It can be regulated, as we're going to get to in a bit, but it is, let's just say, less saturable. That is, it tends to just open up and you have a wide bore tube and a lot of stuff can fly through it. It's not interacting with the individual ion as it passes through. It's just a tube that's opening up. They also, because there's not that much interaction, they tend to be less specific. So you can do experiments like with a sodium channel. Rubidium will also go through that. So it's more of just a charge, and as long as you're not too big, you can fly through it. A transporter is very much of an enzymatic-like interaction with the solute, or with the material being pumped. And so we can establish a Km, we can establish a rate, because we are physically interacting and holding on to that glucose molecule or whatever it is you're taking up and bringing it through. So that is the passive side of things. We'll be getting into channels here in a moment when we get into neuronal uptake. On the active side, we are actively going against the diffusion gradient. That means we're moving molecules from an area of lower concentration to an area of higher concentration. We're accumulating stuff. And this is going to require energy, and so we can then have two different kinds of systems. We have a direct active transport, and this is using ATP hydrolysis. And in fact, I think on Friday I was like, I had to look it up. The sodium-potassium ATPase is the kind of standard go-to for direct active transport, and it in fact is having a phosphate covalently linked to, I think, an aspartate R group. And that causes this dramatic change of shape of the protein as that negative charge plop goes on there. And so there's a whole set of equilibria going on of the shape change that happens when the sodium is binding versus the potassium binding. The main point with the sodium-potassium ATPase is sodium is going out and potassium is going in, assuming the cell is oriented that way. So that is direct active transport. And it is critical because that establishes an ion gradient that can be used for indirect active transport. Indirect active transport, then, is using a co-transport ion, and we'll look at this in just a moment, in that you've got an existing gradient, and you use that energy as that gradient is being resolved to drive the movement of something against its gradient. And we'll see there's a couple of different ways you can play this. So this is a standard kind of breakdown of the different transport systems. And just for the heck of it, we'll throw a couple of figures up. The intestinal lumen is a really great place to look at these different kinds of systems because we pretty much have both active and passive transport systems going on there. Hold on a moment. Here we are. Okay. So, just starting here on this figure, you don't need at all to memorize the relative ion concentrations of different ions in a cell, but there are a couple of things to note that are worthwhile. I'm going to plunge the room into darkness. There we go. So, a couple of points just to keep in mind is that sodium is always going to be much lower inside than outside. I mean, look, tenfold difference. We're talking about a big sodium gradient here. Potassium is the exact opposite. Potassium is always a lot higher inside than it is outside. This is because the sodium-potassium ATPase is running all the time in every cell. And again, as we mentioned, something like 30% of your total caloric intake is being used to drive sodium-potassium ATPase, which is kind of an amazing thing to think about. That's what we spend 30% of the ATP generated for basal metabolism, simply to pump ions around. Magnesium, I don't really care about, but there you go. Calcium is another one that is in vanishingly small concentrations inside the cell. This is a little bit of a misnomer in that it's actually sequestered, and so there's different kind of calcium holding reticuli, the sarcoplasmic reticulum in the muscle cells, and other vesicles in non-muscle cells that have got a fair amount of calcium, but in the cytoplasm it's actually kept very low, and that's because calcium is a real potent activator of a whole range of kinases and different kind of activities. So it is in your best interest to keep a tight rein on your calcium. Let's see, that's really the main things. The hydrogen ion, this is essentially establishing pH. The pH 7.2 or so, sure, physiological saline, we're happy with that. Chloride is higher on the outside than it is on the inside, and there are chloride transporters that are moving things around as well. All right, so, yeah, we'll get into this in a little bit. Now, so this is the difference between a transporter and a channel, passive transport, and there we are. So we've got simple diffusion, channel mediated or transporter mediated, and then we've got active transport, but we also can have indirect active transport. Another thing that is actually part of this whole game is that cells can often be faced with an electrochemical gradient that either you have to overcome or that they can use. And the electrochemical gradient is essentially we can think of it as actually as a voltage, but if you've got a negative charge inside, that's going to have an electrostatic attraction for pulling positively charged ions across it. And so it can add to a little bit of the push of ions in one direction or the other. Yeah. And this is a diagram of something called an aquaporin. Water molecules, depending on the where you look and how you're thinking about it, water molecules can actually move through these different pores. They can also make their way through the sort of the different parts of the phospholipid bilayer. So water can diffuse fairly easily across the plasma membrane. Yep. So lots of different kinds of pumps and things going on here. And okay, so one of the classic places that we look at the effect of maintaining a high sodium concentration outside is glucose uptake. And so we've got a couple of different kinds of glucose transport systems depending on where you are in a cell and what kind of cell type you're looking at. And we'll see this in just a moment. But, you know, the cell's conditions are not the same in all parts of the body. The conditions of the cell in terms of glucose concentration are different in a red blood cell than they are in the gut, the intestinal lumen. And so we have the glucose passive transporter, in which we are driving glucose with its concentration gradient, but bringing it in by interacting with this glucose transporter. We've got a binding site, dumps it into the cytoplasm. Very often, the glucose is going to be modified so it can't diffuse back out. And the other kind of system for glucose transport, and for lots of different things, is using a co-transport system in which we're now talking about an indirect active transport. And the main workhorse for indirect active transport is the sodium-potassium ATPase, i.e., we depend on there always being a higher concentration of sodium outside the cell than inside the cell. And it's kind of like having a base set of assumptions that the cell knows, okay, I can expect that the sodium concentration is going to be higher outside than inside. So I can have some kind of system that is using that sodium gradient, because you can depend on it always being higher. I don't have to worry about that part of the equation. Somebody else is dealing with that, and that's the sodium-potassium ATPase. So this is an example of a co-transport system in which we've got an ion that is moving with its gradient and driving an ion or a molecule against its gradient. This is the ATP-driven pump. And then there are in some blue-green algae and things, a light-driven pump, but they're not in animals, and I ain't got nothing more to say about them. There you go. All right, so this is the principle then behind the sodium-potassium ATPase, is that we're pumping sodium out. That is creating a positive charge out here. We're pumping potassium in, but because we're pumping a lot more sodium out, and the cytoplasm is filled with all these proteins and amino acids that are negatively charged, the inside of the cell, the cytoplasm, tends to be a lot more negatively charged than the outside. So one of the things to keep in mind is that we always have these ion gradients, but we also have this electrochemical gradient as well. So let's see what else we have here. Here we have a dam. The notion being that the accumulation of ions is in fact a potential energy source, much in the way that we've got a dam holds back water and then allows it to pass through, and you have turbines that spin and you make electricity. All right. This is a good analogy for what you can think about is if you've accumulated a bunch of ions on one side of the membrane and now allow them to pass through a very specific little channel, that movement is going to actually drive some energy system. I mean, it's going to be a way of generating energy. Another way of thinking about this is the energy that it takes to accumulate against a gradient, you can actually use that energy in a different form. You can transfer that energy. And so here we are. And we're going to go back to the board here in a moment, but this is more or less another kind of transport system, an active transport system. In this case, it's calcium. So we were looking at sodium earlier. This is calcium. To calcium bind, there is the binding site for ATP, and the direct phosphorylation of the R group on aspartic acid is negatively charged, but now is super negatively charged, and that causes a change in shape. You push something out, it changes the shape, and the calcium has different binding affinities to the protein depending on its configuration. And so when it's open like this, it's actually a fairly low binding affinity, and so it just gets diffused out. All right. So here then is the point. We can use ions that we've accumulated on one side of the membrane or the other as a driving force. And so this is an example of active co-transport or indirect active transport. You're not actually hydrolyzing an ATP directly. You are using an existing gradient. So we can have a couple of different games. There is what's known as symport, in which you've got the thing you're trying to accumulate and the sodium ion, as an example, are both moving in the same direction. You can also have an antiport system, and off the top of my head, and nowhere in the book do they ever list one, I don't know, but there you go. You have a co-transported molecule that is the driving force going out and something's going in. But this middle one, is the symport, is the more standard. All right, so is there anything good here? Yeah, all right, so this is the glucose cotransport system. It depends on sodium always being higher outside. So sodium is essentially the water driving our turbine, and the sodium concentration, a lot out here, it binds to the cotransporter system. It's actually, when the sodium ion binds, it makes us into a very high affinity binding shape for glucose. It snags glucose, it then opens up, and one of the nice things is we've got a sodium diffusion gradient, A, and B, we also have an electrochemical gradient because sodium being positively charged, cytoplasm being negatively charged, we have a charge interaction game going on that makes the sodium move through even higher, and you start back over again. So you're accumulating glucose by using the sodium gradient. A classic test question that I will ask, and many will miss, is describe the indirect active transport of glucose using sodium, and blah, 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 symport, fine.
And then the question is, what's the role of the sodium-potassium ATPase in this process? And people say it doesn't have a role. Like, au contraire. The sodium-potassium ATPase is always maintaining that sodium gradient. So as long as you can depend on sodium being higher outside than inside, this system works. Yeah. This is actually the figure I was driving towards because it's a bit more complex than I wanted to draw. The lumen of the gut is an interesting place to hang out. So this is in the small intestine. Pretty much just a little anatomy, physiology. The small intestine, the duodenum, the jejunum, and the ileum, right below the stomach, they take little packets of food from the stomach, squirt it in, and they begin working on it. So your stomach actually is emptying into the small intestine in little bursts. You don't dump all your stomach contents into the small intestine because there's too much work to be done in the small intestine for it to have a huge amount of material. So the stomach is slowly squirting a little bit of your chewed up breakfast into the small intestine. Now, we've got a reasonably low glucose concentration in the gut lumen. I mean, you've got some glucose from your cereal, but you also have lots of water. Now, in the cell, we're going to end up with a fairly high glucose concentration, and the reason you're eating is that your body is low in glucose relatively, and so we've got this range of glucose concentrations going on here. Now, let's look at how this actually works. We've got glucose being actively taken up from the gut using the sodium co-transport system. In this case, the sodium ion is actually in the gut lumen. The sodium is the sodium that's in your food and probably just in your water. And this is principal then because the cytoplasm is always having its low sodium concentration because of this little friend, the sodium-potassium ATPase, pumping the sodium out. The glucose is brought in, and then we now have a higher concentration gradient here than here, so we don't need to use a co-transport system. It is just simple diffusion. We have the glucose passive transport system passing it out. A couple of things to note. We have these little tack wells between the intestinal cells. These are known as tight junctions. And this is a critical thing. If you were to pierce your intestine, say with a chicken bone, that's not good. One of the ways we work is that essentially, you're a tube, and you've got the outside part of the tube, or a donut, better example, and then you have the donut hole. The donut hole, that is the mouth and intestinal tract up to your rear end. And it is essentially contiguous with the outer surface. And so you need to maintain that integrity. Consequently, we've got these little attachment points between the cells that prevent material from diffusing between cells and then getting into your abdominal cavity. We don't want that. Everything that gets into your body is having to pass through some kind of membrane system. And so unless it's water or some sort of lipid-soluble molecules, sterols and things, it's going to be taken up by some kind of protein system. It's going to be monitored. It's going to be evaluated. And so that means that not everything you eat gets into your body, which is a nice thing. Additionally, you have here bacteria and all kinds of things floating around in the gut lumen that you just as soon not have in your body cavity. So keeping the integrity of these tight junctions is important. I think of it a little bit like mortar between bricks. So there you go. All right, so that's the way we can move stuff through the intestinal epithelium. Another just interesting tidbit here as we're driving over this, because anything interesting here? Yeah. Plants have vacuoles, they can accumulate stuff there. Yeah, you can. Let's see. While we're looking at this, we can just tell a bit of a story.
again, no, don't memorize this. But these are transmembrane pumps, the sodium-driven glucose pump, sodium potassium ATPase, we've already talked about, calcium pumps, proton pumps. Yeah. One of the things we have, proton pumps, hydrogen ion pumps, that are changing the pH in different spots. You got proton pumps that are pumping hydrogen ions into your stomach to increase the acidity of your stomach. We have proton pumps in different vesicles that are pumping hydrogen ions into vesicles to lower their pH. One of the notions is that we have a lot of different hydrolytic enzymes. Actually, turn the lights back on. There are hydrolytic enzymes inside a cell that, were they to be broken loose in the cell, they begin consuming the cytoplasm, which would be a bad thing. They are essentially like a little trash can. And they act and they work because the enzyme systems inside these vesicles they're only active at low pH. So you pump hydrogen ions into that vesicle, drop the pH, these enzymes are now active. If for some reason you take a blow to your kidney, somehow you rupture, that vesicle were to rupture, those hydrolytic enzymes get dumped in the cytoplasm, but the pH is high enough that they're not going to be active. All right, so another interesting point here is that we have inadvertently, as it were, driven over the mechanism of diarrhea. In that, diarrhea can be caused actually by a variety of different things, but things like bacterial-caused diarrhea, cholera, and food poisoning type diarrhea often happens because the large intestine's job is to do nothing but pull water out of the poop to be. So the small intestine is doing all the digestion, all the heavy lifting. Once you make your way out of the small intestine, pretty much you're done. You can take it out and pipe it away. The only thing is, it's got a lot of water in it. And so we want to actually pull that water back into the body. You don't want to dump the water. It's one of the main things that diarrhea can cause, severe dehydration. So in order to pull the water out, what we're going to do is, in the large intestine, we begin pumping salt out of the poop to be. And the main ion that we're pumping out is a chloride ion. And so we have a chloride transporter that's pumping chloride out. There are different bacteria toxins that can inhibit that chloride transporter. And so water follows salt. If you are pulling chloride and salt out of the poop to be, the water follows and is reabsorbed. If you don't, oopsie, you will have now very watery stools. Oh boy. Now, the other side of this is you can ingest a salt that your body doesn't take up very well, like magnesium, and the magnesium, as it's passing through, can't be pulled out. It acts as an osmoticum. It pulls water in, and whoosh, you either have a low-dose magnesium, a magnesium-based laxative, like milk and magnesia, or at a higher dose, you have a way of flushing your entire large intestine out for a colonoscopy. Just a little bit of tidbit now that you know. All right. Now, while we're in the area of ion transport and membranes, it's important that we throw in here the mechanism of neuronal signal transport, in that the nervous system operates by changing the polarity in little patches of membrane. This membrane polarity change happens because of ion concentration change. So we were talking about then a variety of ion transporters. Specifically, I'm just looking to see if there's anything I need to show you here. We've got, and just as a little note here, these are all passive systems. So remember, we had passive systems We could divide it into simple diffusion. We had transporters, and we had channels. And we're going to be talking now a little bit about passive ion channels. Less specific, less saturable. These ion channels, there's this little tube, they actually have some control over them. And most ion channels tend to be what are referred to as gated ion channels. So a gated ion channel, as the name implies, has got a little gate on it. Kind of like a flush valve on your potty. And we got it like this. We got one side and the other, and then some kind of little ball hanging there. And while this is actually somewhat fanciful, the fact is there are pretty good three-dimensional models of these proteins that actually look a lot like this. They're very often 7 membrane pass proteins, so they make a little tube, and then there's going to be a little dangly bit that is moving around and plugging it up or dropping it open. So gated ion channels. The next bit then is what are controlling them. And we've got a range of different gated ion channels. There are voltage gated ion channels. Voltage gated ion channels respond to a voltage, to a very slight voltage change. They can snap open in response to a change in voltage. Another classic gate is a ligand gated ion channel. A ligand gated ion channel is responding to a small chemical, also in the chemistry biz referred to as a ligand. For years I used to mispronounce it apparently as ligand, and then a chemist said we say ligand. And so I say ligand. Ligand. It's a small little chemical thing. Ligand gates, these are responding to a chemical. And then we've got a broader class known as the mechanosensory. And these are responding to either movement or some kind of electromagnetic radiation, for that matter, like your eyeballs. Mechanosensory gated ion channels, they are responding to things like stretch receptors, or a stretch receptor would be a mechanosensory gated ion channel. If you've ever been to the doctor, where they have you cross your knee, and then they take the little hammer here, and your leg shoots out and kicks across the table, it doesn't. You get a little twitch, and that twitch is because that hammer is stretching part of the one of the ligaments, and that ligament stretch is causing this involuntary motion. So it's that stretch actually is causing the neuronal change. Mechanosensory gated ion channels are part of what is responsible for knowing where we are in space. Touch your nose. And so you can know kind of the positioning of your different body parts this way. Also, like the ears, your sense of hearing is happening because there are these little cilia that are moving and deflecting in response to sound waves. As they move, they can open an ion channel. So the voltage-gated, ligand-gated, mechanosensory-gated. Just a little point about a ligand-gated ion channel. Kind of an interesting game here, and you probably have some experience with this. There are a class of thermosensory ion channels. And for whatever reason, who knows evolutionarily why, but some of the thermosensory ion channels are also ligand-gated. And here's the point that I think is kind of cool, is that you have ion channels that respond to change in temperature. That's how you know something feels hot and something feels cold. There are classes of these ion channels that also respond to a chemical ligand. And so that's why you can have capsaicin is the active ingredient in peppers And it tastes hot and feels hot because it's actually binding to one of these thermosensory ion channels. It's a way of spoofing the system. Another one is oil of wintergreen in an altoid. You've got another ligand that is binding to a different thermosensory gated ion channel and saying, hey, this feels cold. And so it is truly a hack or a spoof of your nervous system and that these things work, which I think is just quite bizarre. And it turns out that there are some organisms that don't have the same thermosensory gated ion channel that binds to a ligand, which means that there are guys like little birds can eat peppers that would make you just go to your knees screaming, but for the bird, it's like eating a crayon. There's just nothing to it. There's a little bit of wax. And you may or may not have had the experience of cutting up jalapeno peppers and then touching your face. Or even worse, touching other parts of your body. You talk about, woo, that's hot. And that's not easy to get rid of. WD-40 probably would do a pretty good job. I've not really fully explored that. Because the capsaicin is like a wax and you need to really dissolve that stuff. Anyway, these different kinds of gated ion channels are critical for the transmission of a nervous system signal. And we're going to do a little bit of neurobiology here. Because if not here, where do you get it? So, some basic neuroanatomy, and we're talking really basic. A neuron has a couple of major parts. We've got what's called the cell body, which is where the nucleus is located. And it's got a bunch of cytoplasmic extensions coming out. So the nucleus is right there. And these cytoplasmic extensions are known as dendrites. And then there is generally one very long extension, and this is the axon. So in a giraffe, you can have some axons that are really, really long. I mean, you got axons that are going from parts of your spine all the way down to your pinky toe. So everybody wiggle your small toe. That is a trip from, mid-back all the way down. So it is a long-ass cell. Very long cytoplasmic extension. That is the axon. And then we have the axon terminal down here. All right, and so just to complete this thing, you're either going to have this axon interact with another neuron and dendrites, or a muscle, and we've made the neuromuscular junction, but we'll hit this in a moment. All right, so the mechanism by which a signal is sent down the axon is by carrying a small voltage down the axon. It is effectively a little wire. But now, if we think about like building a little circuit, you could run electricity through that circuit in two different directions. With this system, the electricity, the signal is running in one direction, always going this way. And what we observed, and a lot of this was done back in the 40s and 50s, one of the classic model organisms was the axon from a squid. I don't know any squids. Maybe I've eaten a few squids. But apparently squids have really big axons. I just take it on faith because we always talk about the giant squid axon, or the squid giant axon. It could be from a giant squid, I don't know. Regardless, apparently if you take a squid and you do a very careful dissection, they've got some neurons that are pretty darn big and fairly easy to manipulate. Another classic system has been using something that is known as a patch clamp, which again I've only heard of. I knew a guy that was doing them, and he had some really bizarre looking little tools. A patch clamp is essentially a glass tube. You use a little micro forge, and you pull very tiny glass needles with a little hole in them. And you can take cell membrane and have one of these glass needles sitting on top of the cell membrane. You can actually grab a little piece of it, and you have a wire that's running down a little electrode, these two tubes, measuring the electrical current change. Basically have a little voltometer there, and you can observe changes in the voltage this way. What we see happening is the following. We can have a neuronal signal transferred down the axon, and it manifests itself as a brief, we can think of it as a little voltage spike. It is typically known as a depolarization. Now we're going to oversimplify this a significant amount. In most animals, the axon is actually covered with a tube of insulation known as the myelin sheath. And there's little gaps between the myelin sheath, and this allows for the jump of electrical impulse from one spot to the next. So a degradation of the myelin sheath is a real significant problem because it can lead to essentially short circuits and abnormal movement of the electrical impulses. And the name of the disease that happens when you have myelin sheath degradation just flew out of my head. And I'm not going to bother looking at any notes, but it'll come to me in a moment. I'm thinking like muscular dystrophy, cystic fibrosis, ALS. ALS actually is one of these, is a breakdown, but there are other ones. Crap, it's there. All right, so we're going to simplify those though and look at a depolarization event in the membrane. So we have the initiation of a depolarization event, which we'll actually pick up down here. Let's just go ahead and start. We have a depolarization event. It's primarily working like this. We have sodium and potassium channels that are going to be opening, and chloride channels that are going to be opening and closing. And it starts out like this. We've got, here is 0 voltage. We've got minus 60 millivolts, which is, I don't know, if you've ever taken a 9-volt battery and stuck it on your tongue, that's 9 volts. This is millivolts. It is very, very low voltage. You might be able to light an LED off of it, but not very well. But we are running this kind of voltage in our bodies, which is kind of cool to think about. So minus 60 millivolts, and this is known as the resting potential. So if we were to think of this as in time, this is actually in milliseconds. You have the resting potential. And then you're going to have some kind of membrane depolarization event, an excitatory signal. Membrane depolarization results in the snapping open of chloride channels and sodium channels, such that sodium, which is always higher outside, remember, than inside, sodium rushes in. As positive ions rush into the cytoplasm, the voltage goes up, and it goes up to plus 40 millivolts. At this point, the sodium channel snaps shut, and we've got what's known as a potassium leak channel opens up. Potassium now starts to move from its higher concentration into the cell outside, and that causes the voltage to go down. And actually, we then have chloride coming in, we go below the undershoot, and then we have to kind of work our way back up. So this is what would happen if you were to monitor a patch of membrane on a neuron and you were to give it a neuronal impulse. You flick it. You actually can very easily do this synthetically by adding a neurochemical or giving it a little tiny electrical pulse just to kickstart it. I've never had acupuncture, but I have seen, like in full-blown acupuncture, they put needles into your face, into different parts, actually hitting nerves, and then they apply a very slight electrical charge. And what you're doing is you're doing some funky-ass stimulation of these neurons, given that if you do it that way, you can shoot stuff back this direction. But anyway, it's kind of an interesting game. Now, let's think about what's happening here. We got sodium rushing in, and now potassium moving out. And chloride is also moving back in. At the end of this little pulse, this membrane depolarization, this voltage spike, let's just refer to this as membrane depolarization. At the end of this, the membrane or the cytoplasm ion concentration is completely screwed up. And so the sodium-potassium ATPases have to immediately go back into action, pump all the sodium back out, pump all the potassium back in. And that's what causes what's known as the refractory period. That means there is a window in which your neuron can't fire again. And so this is partly going to be responsible to how fast you can have a neuronal impulse firing down a neuron. It's not just a wire. The membrane itself, or the cytoplasm itself, is being disrupted. Now, another thing to keep in mind is that this is happening in little patches. The membrane doesn't, or the cytoplasm doesn't depolarize whoosh in one long or one immediate pop. It's happening as a wave. And the myelin sheath actually allows for the voltage to jump from one spot to another, but that's not important. So it's not the entire cytoplasm depolarizing. It's patches of it, these discrete patches. And the reason this is cool is that if this patch is going through a depolarization, it is depolarized because this patch just went through a depolarization. So this is where these voltage-gated ion channels are coming into play. A change in voltage here snaps open the change here. That change in voltage here causes that spike to go up. You can't have the signal running backwards because this guy's cytoplasm is completely screwed up and it can't have another voltage change. So the voltage is always moving that way. And it is dependent on each little neighboring spot having this spike, and that causes the next guy's set of voltage-gated ion channels to open up. Let's just take a little quick figure here, and we'll get into one of my favorite aspects of neurochemistry on Wednesday, and that is the ligand-gated ion channels and how they're working. But and just taking a moment here while I'm flipping through some slides and thinking, what is the damn myasthenia gravis? No. Oh, crap. I'm sure it's going to pop in my head as soon as class is done. Multiple sclerosis. Thank you very much. Anyway, multiple people that suffer from multiple sclerosis have a degradation of the myelin sheath. So it's progressive and it gets worse and worse and worse. And you end up having neuronal impulses that are kind of firing in odd places. Let's see. You're right. I don't have any time at all. But just for a second here, just look at the figures. This is a really wonderful story of potassium ion channels that are mechanosensory gated. We'll get to it on Wednesday. Your next equation, who cares? Patch clamps, fine. Team needles, very cool. All right, so this is what's happening, is that you have, move to it. The axon and the squid. And here we have the depolarizing stimulus, minus 60, you pop up to plus 40, and then you go back down. And a good figure to kind of review is boop, 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 boop. This one. Yeah, like this. Where we're looking at the sodium channels and seeing how the propagation, when you have depolarizing events, it pops this one, it causes the sodium to rush in, and then it moves it down that way. Hey, thank you for your attention, and I'll catch up with you later. Ciao.